Thank you, Jeff. Thank you all for coming. My name is Paweł. I run a laboratory of comparative genomics at Virginia Bioinformatics Institute. We do a variety of things, like analyzing genomes of organisms ranging from fruit flies to whales. But today, I'm going to tell you about a major discovery that my lab has recently made. We came across a new epigenetic phenomenon having a large effect at the genomic scale, comparable only with X chromosome silencing in mammalian cells. It's large scale, maybe even bigger than X chromosome silencing in terms of its impact on transcriptional output, because it's affecting more than 50% of the whole transcriptome. It's central to metabolism because it involves ribosomal RNA genes that, by the way, are the most ancient part of all genomes. It involves genomic imprinting, which is the first example of genomic imprinting in amphibians, which are not supposed to evolve, actually, genomic imprinting, because there's no parental care, there's no nursing there, there's no intimate mother-offspring relationship. It's related to this phenomenon that I will be talking about. It's related to hybridization between species, or at least it's more pronounced in hybrid genomes. When species hybridize, two distinct genomes merge, forming de facto a new genome that undergoes dramatic changes and epigenetic reprogramming. These changes may cascade into predictable phenotypic patterns, like those described by Holden's rule that relates patterns of hybrid sterility to genetic sex. 11 years after Holden's rule was formulated, a brilliant Russian cytologist, Michael Navashin, came across another hybrid regularity at the cytological level. He conducted crosses, interspecies crosses between hogs' beards, and found a very interesting phenomenon that he named differential amphiplasty, later renamed to nuclear dominance. So he looked at chromosomes in metaphase and noticed that there is a change to structural features of chromosomes between parental species and their F1 hybrids. So, for example, this is a hybrid between species A and B. And so this feature that I'm referring to is satellite and the secondary constriction. So one of them disappears in F1 hybrid. So this is expressed only from species A, and there is no expression of this feature in copy coming from species B. This is a hybrid BC, so only satellite from species B is expressed, and this feature from species C is not expressed. And same with hybrid AC, only A is expressed, C is not expressed. And it turns out that there's a hierarchy of this dominance, that if species A dominates over species B and B dominates over C, then A also tends to dominate over C. Holden's rule attracted lots of interest in research, but unfortunately, Navashin's observation has been largely forgotten, which is a shame because it occurs not only in plants but also in animals, including Xenopus frogs that we worked on. And in Xenopus, what happens is that parental species express two nucleoli per nucleus. F1 hybrids, like between species like Laevis and mulleri, or Laevis and Boralis, there is only one nucleus formed. So one is suppressed, similar to what Navashin and then others observed in plants. It was believed, so it was described many years ago, and it was believed that this dominance is driven by species, namely X. Laevis dominates over other species, regardless of the direction of crosses, which turns out not to be true. So we re-examined the patterns, and we confirmed that the only thing that we actually confirmed was the number of nucleoli, but all the rest is not actually as it was described or believed. Nucleus is forming around a chromosomal part known as nuclear organizer region that contains ribosomal RNA gene cluster. So each unit or each repeat encodes sequences for 18S, 5.8S, and 28S ribosomal RNA genes, and it also includes a very important intergenic spacer DNA that contains multiple regulatory motifs. Ribosomal RNA, like 18S, is very conserved, but these species are old, and we were able to find species-specific SNPs that could be used as markers to quantify expression of ribosomal RNA. And for that, we used droplet digital PCR assays that turned out to be more effective with these huge numbers of repeats, because there are 500 or more repeats like that in Xenopus frogs and also other organisms like humans. These results were confirmed with other methods like RNA sequencing and next-gen sequencing. So here are the results, quantitative results related to ribosomal RNA expression. So the first cross was between X. Laevis, mother, and X. mulleri, father, and what we saw was X. Laevis expression. Then we did the same thing with Boralis, cross to Laevis. Again, Laevis dominates, sort of as expected from the literature. But then we did reciprocal crosses, and the result is really black and white. So it's not species dominance, this is maternal determination. And then we did back crosses. First back cross to Laevis, mulleri, and Boralis. And all of them had Laevis expression, which maybe is not surprising, because when you think about these mothers actually had Laevis expression to begin with. So it just carries over. But then we did a back cross using mothers that came from reciprocal cross where Boralis was mother. And we found Laevis expression against sort of maternal control that I described earlier. We did also a back cross to Boralis. So basically, there is no Boralis expression in parents, because this is there is no Laevis expression, because here is Boralis expression, here is Boralis paternal, but there is still a slight tendency towards Lewis. So what's going on here? So we think that what really changes back crosses compared to F1 is recombination, which is very important to stabilize the whole cluster of genes. So when F1 hybrid matures, there is recombination, it's germline, and then we back cross this female after recombination with Lewis, and then once Lewis genes recombine into maternal chromosome, it drives expression, it dominates because, and it's pretty well described, or it has been described before, this IS region contains more cis enhancing factors. You may say, okay, hold on, why do you think it's actually coming from maternal chromosome, not paternal, because paternal is also Lewis? So to answer that question, we made a tri-hybrid, so we introduced sort of a new marker from Boralis. So now here is the F1 between Lewis and Mulleray, and we crossed this female with Boralis with the third species to produce a tri-hybrid. Now paternal chromosome contains a different marker from Boralis, but we still see Lewis dominance, which means that it comes from maternal copy after recombination, after Lewis recombines into maternal chromosome. Then question arises how it is related to sex chromosomes and maybe conflicts between sex chromosomes because there's I mean, it's very interesting in the context of genetic conflicts, and we know that sex chromosomes actually a big part of it. So we sex-reversed frogs, and I'm not going to go into details, I don't have time for that. We actually published that in Science a number of years ago. So we created ZZ female. So normally ZZ is a male, but we convert it into a female using a hormonal treatment. And basically these females are, ZZ and ZW females are identical, they produce eggs. Okay, and so we did the cross. We used sex-reversed F1 hybrid, which is a ZZ female. We crossed it with ZZ Mulleray male. We got a backcross zygote, and there was again dominance of Lewis, which means that there's nothing really special, funky about sex chromosomes in this context. Finally, we realized that there is a very remote connection between the number of nucleoli and the pattern of ribosomal RNA expression. It's not all about epigenetics. Turns out that the suppression of nucleoli is related to the number of ribosomal RNA gene copies. So we counted gene copies in parental species. Those are the numbers, they are close to each other, although there is some significant variation. This is what we would expect from Mendelian ratios, and this is what we observed. We observed massive loss of ribosomal RNA gene copies, like 50%, which, and we also see it in back crosses, which really explains why they underdevelop nucleoli. And there's another thing very interesting, which is shown here. There's underrepresentation of red color, which corresponds to Lewis. So Lewis is sort of preferentially lost in the whole process. It doesn't make to F1 zygote for some reason. So on one hand, Lewis is good in driving expression because it dominates in expression, but it gives some disadvantage during F1 zygote formation. So to conclude, there is maternal control of ribosomal RNA expression in F1, back crosses and hybrids, a new dramatic epigenetic phenomenon. Once RNA gene copies with longer IS sequences recombine into maternal NORs, they escape silencing, pardon me, and dominate in expression. There's a reduction in the number of nucleoline hybrids, likely as a result from loss of ribosomal RNA genes, rather than some epigenetic phenomena. And finally, in F1 hybrids, X-Lavis ribosomal DNA tends to be preferentially lost during a zygote formation. Thank you. I'll be happy. I don't know if I have time.